Hi, hello. I'm here with another article on endophenotypes in schizophrenia, digging deeper to identify genetic mechanisms. So that is the topic of this article, and it's also my first time reading this article. I'm curious how and what it really mentions. So from this title, we can tell that they're they aim to identify genetic mechanisms related to endophenotypes in schizophrenia, and it sounds like a review study. And schizophrenia is a severe psychotic disorder that is highly heritable and common in the general population. Genetic heterogeneity of schizophrenia is substantial and with contributions from common, rare, and de novo variants, in addition to environmental factors. So it is obviously interesting, and large genome-wide association studies have detected many variants that are associated with schizophrenia, yet the pathways by which these variants influence risk remain largely unknown. So they have a different symptom severity, and it's quite complex, and uh, it makes it very complicated to identify these variants. Variants, and however, the underlying brain dysfunctions forms a more stable trait marker that quantitative neurocognitive and neurophysiological endophenotypes may be able to objectively measure. Okay. And furthermore, many endophenotypes are translational into animal model systems, allowing for direct evaluation of the neural circuit dysfunctions and neurobiological substrates. So they review a selection of the most promising schizophrenia endophenotypes, including propulse inhibition. So there are many studies on propulse inhibition, schizophrenia, depression, mismatch negativity, oculomotor antisaccade. They all must have something to do with cognitive skills, letter number sequencing, and continuous performance tests. We also highlight recent findings from large consortia that suggest the potential of genes, particularly in the neuroglin and glutamate pathways in several of these endophenotypes. So it's interesting. So they mentioned the genetics of schizophrenia here. Genome-wide association studies are very popular. The field of psychiatric genetics shifted to genome-wide association studies in 2007 with the introduction of microarray-based genotyping and the culmination of efforts to map the structure of the human genome. Such studies rely on allele frequency differences for single nucleotide polymorphisms in large samples of unrelated schizophrenia cases and controls to implicate genomic regions containing risk genes. So I think there must be a lot of, there are a lot of studies you can find and see the results. And it seems to be like here, there are many patients and controls. It says produced no significant findings yet. Follow-up studies implicated the MHC and variants in the neurogranin and transcription factor for genes. Another study demonstrated that common genetic variants in aggregate may accounted for 30% of the risk for schizophrenia, and it's also important to check which method they have used to explain and account for the risk, because if they use different methodology, then the numbers do not mean much, and how they calculate it, what are their methods and sample size, or there are many other important information we should include to understand and here next generation sequencing studies. Another strategy in the genetic investigation of schizophrenia has been the application of next generation sequencing method that were pioneered in cancer research. Contrary to the genome-wide association studies focus on common variants, these methods seek to identify copy number of variants and but have so that single nucleotide variants that occur in less than 0.1% of the general population but that have elevated frequencies and functional roles in schizophrenia. So that's interesting. And so, and other one, gene expression studies. Human genome is made of DNA that is organized into 20,000 to 25,000 genes. Nearly every human cell contains the same genes, but different cells and tissue have different patterns of gene expression. These differences in gene expression may contribute to schizophrenia, and so that's also they mentioned the related factors before each gene is expressed. So these transcriptions, which are collectively referred to as transcriptome, control the function of cells. Several groups have explored the use of peripheral blood gene expression signatures and used transcriptomes to investigate the biological consequences of schizophrenia-associated genes. These studies have suggested a potential role for ARC NMDA receptor synapse and immunity dysfunction and central nervous system development in schizophrenia. Okay, it seems to be the article and also mentions the tests, I guess, and there must be a conclusion for this. It seems to be very long paragraph and very long. There are many pages in this article. Using endophenotypes to dig deeper, going beyond the broad clinical diagnostic category of schizophrenia will facilitate the identification of pathways leading from genetic variation to brain dysfunction. A better understanding of the genetic substrates underlying deficits in key domains defined by schizophrenia-related endophenotypes will provide a molecular roadmap to guide the development of novel pharmacological interventions aimed at improving real-world functioning as well as information to guide precision-based treatment decision. And thanks for watching this video, and I wish you enjoyed this article and the way that I mentioned, I present the article, and wish you have a beautiful day. Bye bye.